Right. So now we're getting to the sort of section where we start talking about methods that can be used to sort of give more like sort of biological insights or for more biological analysis of your data. And the first thing we're going to be talking about is clustering. So clustering is very useful because it allows us really to get to one of these really central questions in biology, which is sort of how to define a cell type. So traditionally, sort of cell types were really defined based on the morphology, but then people sort of started to sort of finding these different types of protein surface markers that they could measure, and that allowed you to get sort of a more molecular based definition. But then, of course, with single-cell RNA-seq, you can take that even further. Instead of just looking at a few surface markers, you can look at the entire transcriptome, and then you can have sort of like a bottom-up type of definition of what a cell type it is, and it can be defined based on the transcriptional profile. So this is, of course, sort of very attractive for trying to be a little bit more rigorous about how to define cell type, but it's of course not without problems from a computational point of view. So what this really means, computationally speaking, is that we want to do unsupervised clustering, and this is again a problem that's been around for a very long time in the statistics and the mathematical literature, and it's quite challenging because we're just given the data, and then what we have to do is to figure out sort of how many clusters there are and where are they, where should the different points go. So there are several different strategies out there. The oldest one is k-means, which is sort of quite intuitive. So you assume that there are sort of k different centroids, and then that you try to group the points in your data set to these centroids. So this can be done computationally very efficiently using what's called the Lloyd's algorithm. It scales quite well with the size of your data set. So that's why it's an attractive strategy. It's also sort of intuitive to understand what's going on and what the algorithm is doing and what it's finding. The method one method which is based on k-means clustering, which is used in single-cell RNA-seq, is called single-cell consensus clustering. So what this method essentially does is that it runs k-means clustering sort of multiple times using sort of slightly different input parameters to how it's doing the clustering. And then it takes all these different clusterings. So each of these different parameter combinations, you can think of it as it provides a different solution for how the cells should be grouped. And then it takes all these different solutions and it combines them into a consensus, and then it sort of gives that as its final result. So the main advantage of this is very robust, and it's also quite remarkable that the consensus that you'll find is often going to be better than any of the individual solutions that you have. So this sort of wisdom of the crowd types of approach works really well. The main drawback of this method is that it's very slow, and you can't really use it sort of reliably for data sets with more than 1,000 cells. So another sort of type of like generic clustering strategy is called hierarchical clustering. So this is not very popular in the single cell field. I think the main reason is because it's quite slow, so it scales quadratically with the number of cells that you have. One major advantage of hierarchical clustering, however, is that it doesn't give you just one solution, but it gives you multiple solutions. You can you can get this type of tree here, and you can and then once you have done that, you can sort of choose where you want to cut in the tree, and that will give you the final outcome. The type of strategy which is most popular these days are these graph-based methods. So these were sort of really emerged in the noughties and in the last decade, and they were really devised for sort of looking at these very large internet or World Wide Web related data sets. So the main advantage of these methods is that they scale very well, and they can handle sort of millions of nodes or millions of cells at a time. So The most popular method is known as the Louvain method, or Louvain clustering. And what it will do is that it tries to sort of maximize the modularity. So modularity is modularity is defined as a sort of a set of nodes that have a lot of internal connections between themselves and very few connections to the other nodes. So it tries to sort of optimize this quantity, and then it does so in a sort of hierarchical fashion. And again, the main advantage is that it scales very favorably. It's n log n, and it can be run on these really large data sets. So one big challenge of clustering is that we typically don't really have a ground truth. So it's very difficult to sort of evaluate to what extent the clustering is sort of correct. So there are sort of various heuristics that you can use to, and what they typically do is that they look at sort of how tightly are your points sitting next to the centroids or cluster centers that you had identified. So my favorite method for doing this is the silhouette score, and it's quite easy to interpret, and you can sort of calculate one silhouette score for your entire sample, which will tell you sort of how good the overall clustering is. But then you can also calculate for sort of the individual clusters to sort of identify which clusters are the ones that are sort of performing poorly, and where you might have been able to do a little bit better. So another important thing to say about clustering is that it's typically the case that there is not a single correct answer. So you can, for example, imagine if you have a data set from say a cortical brain region, then you could cluster that on multiple different levels. So you could for example, you could have, on the one hand, you could have like very high level clustering, so where you distinguish the neurons from the astrocytes and the microglia. And then you could imagine a sort of a more fine-grained clustering where you neurons are split into the excitatory and the inhibitory neurons. And then you could think of it at an even finer level where the neurons are sort of split into the different layers where they would come from. So all of these solutions would be correct in some sense, and which one is sort of best is really determined by your question and by your application. And I think my sort of advice and recommendation is for most applications, it makes a lot of sense to sort of consider clustering at sort of two different granularities, and then you want to make sure that sort of the results that you sort of get from one granularity is sort of robust in the other one as well. So that's often a useful way of working with this. So one important sort of caveat to say about pretty much all of these methods I've been talking about before is that they tend to perform well for the large clusters, but they typically have a really hard time for very small clusters, which would of course often correspond to like rare cell types. So the reason for this is sort of intuitively, it's not hard to understand, because if you have a really small cluster, it's really hard to know if this is sort of a real sort of biological outlier or if it's just some sort of noise or sort of technical outlier. So if you know that you have or suspect that you have these sort of small clusters, so for example some type of stem cell or something, then it is often a good idea that to use a different method than the clustering method that I just talked about for trying to look at them, because there are these methods that are specifically designed for identifying these rare cell types. So one example is this RacelD method, which is sort of based on k-means clustering originally, but then it has sort of special extra steps that allows it to more sort of carefully look at the outliers and make sure that they are actually correspond to sort of real biological clusters and not technical noise. So finally, a few words about clustering to think about, because it is a very important step which is part of this analysis, and you will sort of because a lot of the sort of what happens downstream is sort of heavily dependent on how you decided to group your cells. So it's in many projects, this is sort of where you'll be spending a lot of time thinking about which clustering solution is the correct one. And so some points to think about. So one is that, in my experience, in general, clustering tends to work really well, even with very sparse data. And sort of intuitively, I think about it as, if you only have a small number of cells, then, even if you only have a small number of reads, a small fraction of the genes detected, really most of the information is sort of in the presence of absence of the gene. And that will allow you to do really well, even if you have sort of, say, fewer than 1,000 genes detected per cell. Another thing to keep in mind is that there is a sort of dichotomy between the sort of mathematical and biological considerations. So from a mathematical point of view, the sort of the clustering problems sort of very well defined, and we know that there sort of exists a global optimum and so on. It might be very hard to find, but we can sort of, through various means, we can sort of convince ourselves that we've reached it. Unfortunately, sort of the mathematical optimum may not sort of correspond to the most appropriate biological solution. So it is important to sort of take both of these aspects into consideration and really think about how you interpret the solution biologically. And for those reasons, perhaps you want to choose something which is sort of suboptimal from the mathematical point of view. And again, sort of related to what I was saying before, that sort of it works. So these methods work quite well, and you can cluster even very large data sets in sort of relatively manageable amount of time. But what you'll typically spend much more time doing is sort of annotating these clusters and understand what they represent. And today I would say that this is really the biggest bottleneck. So next I'm going to talk about some strategies for trying to sort of understand these different clusters and sort of just annotating them. So one concept which is sort of intuitively very easy to understand is marker genes. So marker genes are defined as genes that are highly expressed in one cluster and lowly expressed everywhere else. So this is sort of illustrated here in this heat map. So here on the rows, you have different types of genes, and in the columns you have cells which correspond to different clusters. So you can see the diagonal here, which corresponds to the marker genes. They are sort of highly expressed in one cell type and low pretty much everywhere else. So these are typically very useful. If you find those, you can go into the literature and try to understand sort of what each of the different clusters that you have identified represent, because there is a lot known already about different markers corresponding to different cell types. Pretty much all clustering packages will get you a list of marker genes for each of your clusters. There are, of course, specialized methods for finding marker genes. One such method is called Comet. And unlike what the most clustering packages do, which is just look at each gene individually, it sort of uses a combinatorial approach to sort of try to find the best set of genes that will represent each cell type. So, of course, there are sort of on the order of 20,000 genes in the human and mouse genome. So looking at all combinations of pairs or triplets is not possible. So what they do is that they have a heuristic, which is quite straightforward. They rank the gene based on their sort of marginal performance, and then they take the top of that list, and then they look at all combinations in the top, and that sort of obviously saves them a lot of computational time, and it seems to be working very well in practice too. So another approach that you can use in order to annotate your clusters is to sort of try to take advantage of other data sets. So in particular, now that there are these sort of various atlas data sets that are starting to come out, what would be really useful is if we could sort of try to leverage what is in there and use that when we try to annotate our newly collected data sets. So this is when you sort of try to essentially map your existing data set to an atlas or to some other existing data set in order to figure out what it best matches. So the first method to do this is called Scmap, and it takes a sort of a very simplistic approach. It essentially it thinks of each of the existing clusters that you had in your atlas or in your reference as a centroid, and then it for each of your new cells from your query data set, it tries to sort of just match it to the nearest centroid. And what it sort of does in order to do this successfully is it uses sort of feature selection. It uses this dropout based methods that I told you about earlier. So so based on that, it only looks at a subset of the genes and calculate the distances to these centroids and matches it to the closest one. This type of problem is sort of quite well set up for sort of a lot of other machine learning techniques, and you'll find many other methods out there. One example is SingleCellNet, which is based on random forests, and it sort of does pretty much the same thing as Scmap does. All of these, both of these methods and many others are sort of unsupervised methods that they trying to learn how to best project the data sets. There is, of course, as you know, a lot known about various marker genes and so on for existing cell types. So it would be, of course, useful if we could take advantage of that information when we try to do this projection or mapping. There are methods for doing that. So those I label as supervised projection methods. And one such example is Garnett. And what it does is that you'll have to give it a sort of a hierarchy of the cell types that you think you have in your sample, along with a set of marker genes for each one of those. And then it sort of uses that and looks at the data, and it tries to build a classifier that will tell you which cell type the cells in your queries sample matches. So we talked about visualization before, and it's pretty tricky for these high dimensional data sets. It will obviously be even trickier if you have two different data sets that you, so one of is your atlas and the other one is your query data set. So one neat way of sort of trying to represent these types of mapping is using these Sankey diagrams. So here I'm showing you one example. So what we have here is sort of on the right, we have the reference, and on the left, we have the query data set. So in both of these cases, the data sets had been annotated beforehand, and then we used the method to map them. So what you can see here is that most of the cells that were labeled beta cells in the, so these are from a pancreas data set, maps to beta cells in the reference data set as well, and a few of them go other places. What's important to point out here is this unassigned category, and this is sort of course not in the reference, but it's something which is very useful to have for your mapping algorithm. So what this really means is that it means that the cells that it found that were given to it for your query data set do not match anything that was present in the reference. So this is essentially a way for the algorithm to say, sort of, this is something that I've never seen before, and this could be the way that you will discover sort of new cell types that were present in your new sample, but not in your reference. Something which is sort of related to these types of questions about marker genes is differential expression. So this is arguably one of the most important applications of bulk RNA-seq to sort of identify the set of genes that are different between two samples. In the single cell world, it's actually more complicated than in the bulk RNA-seq world, because what you typically the groups that we want to compare are not defined a priori, because what we typically do is that we first do unsupervised clustering to identify the groups, and then we start comparing them. And what that means from a sort of statistical point of view is that that means that we'll get more, if we just run the sort of same old differential expression test, we will get more differentially expressed genes because the clusters are, of course, based on on differences in gene expression. So that will result in sort of more differentially expressed genes detected. So that's something which is important to keep in mind when you're doing this. So there are lots of methods for doing a differential expression for bulk RNA-seq. There are also lots of methods that have been designed specifically for single-cell RNA-seq. What's interesting in this is in this big benchmarking studies that was done a few years ago, and they tried to compare these. What turned out was rather surprisingly, I think, at least from my point of view, is that the one method that performs really well, and you can sort of see here by blue being that it does well and red meaning that it does poorly, is just the standard Wilcoxon rank test. So this is a non-parametric test, so it's very easy to use and it's very robust, and so it has really become the standard to use in the field these days, and that's what most people, including myself, would recommend that you use for differential expression in single-cell RNA-seq data. One problem that you often have when you're trying to do this differential expression analysis is that you will end up with a lot of different comparisons, especially if you have sort of multiple data sets from different conditions that each have been clustered, so you have multiple groups. Then you can end up with having a very large number of pairwise comparisons, which will be sort of problematic in terms of cognitively for you in order to be able to process, but also perhaps more importantly from a statistical point of view, that you'll have a lot of multiple testing correction that can sort of result in you losing power. Fortunately, there are methods from statistics to deal with these types of situations. And if you're faced with this type of complex experimental design for doing differential expression, one method to consider is called Muscat, which sort of sets up uses a generalized linear model framework in order to try to be able to group samples and in sort of in meaningful way, and it will give you better power. So I think I will stop here. Okay,